I'd like to now talk about one more feature of prokaryotes before moving on to the eukaryotic cell, and that is the motility afforded some prokaryotic cells by their elaborate flagella. If we look at the flagella found in prokaryotic cells, we find that those are used for locomotion, and prokaryotic cells, we think of them as primitive, but in fact they're actually quite complex. They can chemotact. They can sense noxious substances and swim away from them, or on the other hand, they can detect food sources and swim towards those. So they're quite sensitive cells. They have quite a little sensory system. And the flagella propels the cell forward towards food sources or away from noxious substances. And if we look at the flagellum, we find that it's a complex molecular motor. It contains a protein filament joined to protein rings inside the cell wall. So we have, here's the peptidoglycan portion of the cell wall. Here's the outer portion of the cell wall. And here is the plasma membrane. And the driving of the flagellum, the twisting of the flagellum, is driven by these molecular rotors, really. They're motors that twirl. And they twirl based on the flow of hydrogen ions across the plasma membrane. There's a higher concentration of hydrogen ions outside the plasma membrane than inside the plasma membrane. And as these hydrogen ions flow through this protein complex, that causes a spinning, which then drives the spinning of these structures, which then twist the filament of the flagellum, and that causes a wave of rotation to move down the flagellum and propels the bacterium forward. Now let's tackle eukaryotic cell structure. So let's look at the eukaryotes and the differences between the eukaryotic cell and the prokaryotic cell. So some major features of eukaryotic cells are that they possess a membrane-bound nucleus. This is a double lipid bilayer. That is, it's a double membrane, double phospholipid bilayer. They are more complex in the sense that they have many more compartments in their cytoplasm than prokaryotic cells have. And we'll be visiting those different membrane-bound compartments. So we have organelles and we have the endomembrane system that are part of that constitute those compartments, and we'll be talking about those in great detail. In addition, eukaryotic cells possess a cytoskeleton for support and to maintain structure in the cell. Also to provide changes in cell shape and to drive the separation of chromosomes in the process of mitosis, as we will see. And the cytoskeleton, as we'll see, has three major components. It has actin microfilaments, tubulin microtubules, and a class of cytoskeletal proteins called intermediate filaments that we'll be talking about. So if we take an overview of this and look at a few of these things in overview, we see immediately that the eukaryotic cell has some features that the prokaryotic cell does not. And let's look at some of those features right now. So here is the nucleus, which compartmentalizes the genetic material, the chromatin, from the rest of the cell. And this is, as we'll see, is a double membrane system full of nuclear pores, studded with nuclear pores, through which molecules pass in and out of the nucleus as part of the communication with the cytoplasm. There is an endomembrane system that includes something called the rough endoplasmic reticulum, which is studded with these little brown dots here. Those are ribosomes. And the smooth endoplasmic reticulum, which, like the rough endoplasmic reticulum, is a membrane-bound canal system through the cell, but it lacks the associated ribosomes that are found studying the rough endoplasmic reticulum. There is an apparatus called the Golgi apparatus, also membrane-bound, that is the trafficking center of the cell in terms of membrane components and other components, and it serves as a way station for molecules that are going to be exported by the cell and sometimes brought into the cell as well. We have mitochondria. We have numerous mitochondria. There are thousands per cell, per eukaryotic cell, and these are double membrane bound. Well, they are the sites where oxidative phosphorylation takes place in the production of ATP, the energy currency of the cell. So really the mitochondria are the energy factories of the cell, and we'll be talking a lot about them. There are other membrane features, something called a lysosome, which is involved in degradation of proteins or foreign substances. We have a peroxisome, which is involved also in degradation of certain molecules. And we have elements of the cytoskeleton. Here we see two centrioles that are involved in the organization of long cytoskeletal cables called microtubules, composed of the proteins alpha and beta tubulins. And we'll be talking about those in addition. Here are some actin microfilaments. These are part of the cytoskeleton that consist of the polymerized molecule actin, the polymerized protein actin. And then we also show an intermediate filament, another cytoskeletal element as well. Underneath the nuclear membrane is a meshwork, a network of intermediate filaments made out of the protein lamin, L-A-M-I-N, and we'll be talking about that as well. That has an important role in controlling the structure of the nucleus. And there are free ribosomes studded throughout the cell, found throughout the cell, not just on the membrane of the rough endoplasmic reticulum. And as you know, ribosomes are the sites where protein synthesis takes place. They are the protein manufacturing sites of the cell. Often the surface of this cell has extensions called microvilli. Usually the cell isn't just a smooth surface. Rather, there are membrane projections which increase the surface area of the cell, and we term those projections microvilli. So we'll visit a lot of these features as we go on in this lecture. Now we can move on and look at the features of the plant cell. Many of the features in animal cells are shared by plant cells, with some major notable differences. One, there are double membrane-bound organelles with membrane-bound internal structures in them. These are the chloroplasts. So there are actually 3 membranes represented in the chloroplasts, and they are the sites of photosynthesis. That's how plant cells, that is where plant cells manufacture sugars and carbohydrates from using the energy of sunlight. They capture sunlight in photosynthetic pigments, as we'll study. They have also, plants also have large membrane-bound organelles that we would call vacuoles, and those vacuoles provide a variety of functions, including regulation of cell turgor, water balance inside the cell. So these are the vacuoles, and usually there's a large central vacuole in a plant cell. In addition, plant cells have cell walls outside their plasma membranes, and those cell walls are made of the carbohydrate, the polymerized carbohydrate cellulose that we talked about earlier. So they have cellulose cell walls. Now, in reality, things are a bit more complicated than we draw them in these simple schematic diagrams. The cytoplasm really is a semi-fluid matrix, and this is an artistic rendering by David Goodsell of the Scripps Institute, and he does these wonderful paintings where he takes molecules of known structure and paints them according to their shapes, and then includes in his watercolor paintings the approximate densities, the approximate concentrations of these molecules in a cell. So this is just a tiny slice of a bacterial cell. Here is the bacterial flagellum, and you can see that the cytosol really is packed with molecules. It's not just an empty bag of water, and it would be foolish to think of it that way. So the number of interactions that are going on here are astronomically large. Here is shown numerous known proteins. Here is the bacterial DNA wound around certain structures. So things are a bit more complicated than we tend to draw them in our introductory biology classes. So let's move on and talk about details of certain organelles inside eukaryotic cells now. And let's start with the nucleus. We know that that is where the genetic material is stored, and the nuclear envelope is the 2-phospholipid bilayer membrane. So the nuclear envelope, or the nuclear membrane, consists of, it's a double membrane. It consists of 2 phospholipid bilayers. And DNA in the nucleus is organized with proteins, and we call that complex of DNA and their associated proteins chromatin, which is Greek for colored stuff. So chromos means color, tin stuff, so colored stuff. And it's called that because when cytologists first started using organic dyes to stain cells, they came across ones that lit up the nucleus, that bound the nucleus. So the nucleus was brightly colored with these dyes. And at that time, and this is back in the 1800s, no one really knew what that colored stuff was, but so they call it colored stuff, chromatin. And we know that those dyes were binding to DNA now. We now know that. So here is the schematic of the nucleus. We see the nuclear pore complexes, which are large macromolecular assemblages of numerous proteins that punch holes in the double membrane system and allow mRNAs, for example, to be exported through the nuclear pores into the cytoplasm, transcription factors which regulate DNA to come into the cytoplasm to come into the nucleus from the cytoplasm, where they are manufactured on ribosomes. And inside the nucleus is a structure called the nucleolus, which contains the genes that encode ribosomal components, like ribosomal RNA. The genes that encode those ribosomal components are found in a structure called the nucleolus. So there's quite a bit of nuclear organization that occurs. Here's a detail of the nuclear pore, and you can see the outer and inner membrane, the double membrane system of the nuclear envelope. Of course, that is a double phospholipid bilayer. The nuclear lamina contains the protein lamin that I mentioned before, that's instrumental in providing structure to the nucleus and is instrumental in breaking down the nuclear envelope when the chromosomes need to be segregated in mitosis, when the cell divides. Now here's a freeze fracture view under the electron microscope of the surface of the nucleus. And when you do freeze fracturing, as shown here, you freeze the tissue and then you impact it very hard with metal. Usually you slam the tissue with a piston, and that cracks the tissue along membranes so that you can kind of peer into the membranes. And we see that here's the inner nuclear membrane, and we see numerous nuclear pores, and we can see that those pores are studded with large assemblages of macromolecules of proteins. And here's a cross-section of that under the electron microscope, falsely colored to distinguish cytoplasm from the nucleus. And here is the chromatin inside the nucleus. Here is a nuclear pore. Here are the double membranes, either side of the nuclear pore. So the membranes are kind of folded around that. There's the opening of the pore, and out here is the cytoplasm. Underneath the nuclear membrane, as I've said, is a meshwork of the protein lamin, which is an intermediate filament, part of the cytoskeletal class of proteins. And as I've said, that meshwork of lamin provides structure to the nucleus and is involved in the nuclear envelope breakdown in mitosis. All right, let's now consider the evolution of eukaryotic cells and focus at first on the evolution of the nucleus. So let's have a little diversion here and talk about that.